Okay, so let's continue with our immunity discussion. So we already learned about antigens as essentially name tags on a cell. And we learned about epitopes as a specific region on an antigen. And we learned about paratopes as the thing that, say, a T cell will use to interact with the epitope or to read the epitope in order to decide or to be stimulated to do what needs to be done. So T cells are not the only things that have paratopes. Antibodies and B cells also have these paratopes that are able to read the epitope. And remember, they'll be specific for their particular epitope because they've been programmed through various means, whether it's through cross-reactivity and mutation, or through something called a naive T cell, which is a T cell that hasn't been programmed yet. It hasn't had its epitope programmed, and these would tend to be found in your lymph nodes, and there are things called dendritic cells that can present antigens to those naive T cells and program them, which we'll get into in a couple of minutes. But I first want to get into how the T cells react to various epitopes and the reason why they would react that way. And we need to look at something called an MHC marker. And what an MHC, what that stands for, is major histocompatibility. I'm not having luck with spelling today. Marker, or complex. So major histocompatibility complex, or MHC. So what that word means, histocompatibility, compatibility is obviously like how things get along, if they are compatible with each other, if they belong together. Histo means tissue. So is it a tissue that's compatible with your body, essentially? So if it is a cell that is yourself, yes, that is compatible with your body. If it is a pathogen, a virus, no, that is not compatible with your body. So these MHC markers are ways that cells have of communicating with your T cells, letting them know if they're okay, or if they're infected, or if they're presenting an antigen. So they've phagocytized a virus, and they're saying, hey, we've got this virus here. So let's look at a normal MHC. So this is a cell that is just normal, healthy, it's uninfected by any kind of virus. Your T cell is going to kind of go along here. Its little paratope is going to see no viral epitope on here, and it's just going to hopefully pass on by. It's not going to react in any way. You don't want it to react in any way. You want to leave the cell alone so it can keep doing whatever it does. Now let's say the cell gets infected. So virus gets inside the cell and it starts, using the cellular machinery to make new virus. That little virus epitope ends up in the MHC marker, so up there. So I use that same little crown shape to be the epitope here. So now we have this cell that's infected. It's just essentially a little virus factory. So the T cell comes along and it has its paratope on the CD marker, and that matches this one. And it can react. So it binds there, and that causes a reaction. So we'll see which way it reacts and what this particular T cell is that can react to this class 1 MHC. Our other option is that this might be a white blood cell here, like a monocyte or a B cell, and it's phagocytized a pathogen, and it's digesting that virus. So it's just got I just put half of the virus there because it's half eaten up. But while it's digesting that virus, that viral epitope ends up on its MHC. So it's presenting the antigen. It's showing like a wanted sign, like, hey, have you seen this virus? There's this virus loose around here. So look for it. And the T cell comes along, a very specific kind of T cell, and its paratope is specific for this epitope, and it can bind here, and it can react. So we don't want to react here. We do want to react here, and we do want to react here, but we want to react in different ways. So this is an infected cell. This is just making more virus. We want to kill that cell. So there is a very specific T cell here, which is a CD8 T cell. So remember we said that the T cells could be either CD4 or CD8s. The CD8s react to the MHC1, and you want to kill that cell. So when this paratope binds to the epitope, this T cell is going to differentiate, and it turns into a killer T, which is also called a cytotoxic T cell, and then a memory T cell. Now the killer T cell is going to do exactly what you think it does. It's going to kill this cell using cytotoxic enzymes. The memory T cell is just going to kind of go and remember this specific epitope, this specific paratope here, and it's going to be ready to react if it sees this again. It can turn into a killer T very quickly, much faster than this original process, if it sees this particular pathogen again. Now, in this case, we do not want to kill our B cell or our monocyte for doing its job. We want to help it do its job. So this will be a CD4 cell. So we have a CD8 over here to kill the infected cell. And over here we have a CD4 cell to help the cell that has phagocytized this pathogen. So the CD4 cell, when its paratope binds to that epitope, is going to differentiate into something called a helper T cell, which will send out cytokines, and specifically interleukins, which help to stimulate B cells into differentiating into producing cells that make antibodies. And then a memory T cell, which can quickly turn into a helper T cell if exposed to that same pathogen again. So for an MHC1, the CD8s respond, turn into a killer T cell and a memory T cell. For MHC2, the CD4s respond, turn into a helper T cell and a memory T cell. Okay, so here's cross-reactivity again, just in case you didn't get it the first time. Let me make this a little higher. So just to kind of hammer this point home, you've got this as your T cell here, or a B cell. You've got the virus. This is virus number 1, this is virus number 2. You can see that the epitope is a little bit different, but also a little bit similar. So these are related viruses. This T cell and this one, we'll just call it a T cell, could be a B cell, but the paratope is specific for virus number 1. It fits neatly into that epitope there. So it causes a reaction when it binds. It does not fit as neatly into this one, but it does fit kinda. It fits enough to stick. It fits enough for something to happen. So it'll have a weaker response, but a response nonetheless. And it can slowly, or quickly actually, not slowly, mutate as it differentiates. We start to get more and more specific until finally you have T cells that have a paratope that looks like it fits this particular epitope instead. And that helps you develop your immunity for something. Now, I say quickly, and I said slowly, I've said it all. So the process, it can take a little bit of time. So we're talking just a couple days. So that's not slow. It's also not like immediate. So it's like, you know, you get exposed to something and you feel kind of sick for a little while. That's when you haven't finished this process yet. You're just trying to get your T cells kicking and your B cells accustomed to this new type of virus. And then, you know, you get that moment where it's like the turning point in the sickness and you're like, okay, I'm starting to feel better. That's because this has happened. Okay, so we all know, you know, it takes a variety of times depending on how aggressive the pathogen is, how aggressive your immune response is, how healthy your immune system is. All sorts of things can make it take a varying amount of time. Okay, now making antibodies. Remember the class 2 MHCs. Those were the B cells or the macrophage that were essentially presenting an antigen to a T cell, asking for help in dealing with this particular virus. So here's our B cell, say, and it's digesting the virus and it's displaying that viral epitope presenting that antigen on a class 2 MHC. So the class 2 MHCs are responded to by the CD4 T cells, which we just saw. And that CD4 T cell, if it has that paratope that's specific for this particular virus, it will bind and that will trigger the CD4 cell to differentiate into a memory T, which will, you know, be able to remember the process and differentiate into a helper T if need be, on a later date if we are infected again. But what we're really looking at here is when this CD4 cell differentiates into a helper T. And that helper T secretes those interleukins, which go into the B cell, and that triggers something called B cell sensitization. You can think of it as B cell activation, because you're activating the B cell into differentiating itself. And what that B cell will differentiate into are plasma cells. Plasma cells then make the antibodies, which are released into the blood and do various things, which we'll get into in a minute. And then also memory Bs that can quickly turn into a plasma cell if you are reinfected or if it sees that virus again. So the B cell phagocytizes the virus, displays a class 2 MHC with that viral epitope, a CD4 T cell whose paratope matches that epitope, reacts to that, differentiates into a helper T and a memory T. The helper T releases interleukins. The interleukins cause that original B cell to differentiate into a plasma cell, which makes the antibodies, and a memory B, which can turn into a plasma cell if it's exposed again. So what do those antibodies do? There's a variety of different things that the antibodies can do. And we're just going to get sort of a brief rundown because there's many different subtypes here, and they all do very different things, or, well, not very different, but they all have subtly different things that they can do. But the main types are G, A, M, E, and D. And then there will be little subtypes within each of those. So each one will start with IG for immunoglobulin. Globulin, that's a type of protein, and immuno for immunity, so it's a protein, a globular protein for immunity, and then this letter type is going to essentially dictate how this antibody is going to act, generally speaking. And remember, we can find varieties of each of these that are going to react in a different sort of way, but just generally, on first exposure, you'll very quickly express IgM and IgD. And this is easy to remember because when you're first exposed to what's called a novel pathogen is one you haven't seen before, one you haven't been infected with before. So it's a brand new pathogen. And we all are experiencing a novel pathogen right now, which is why we're having class online at the moment. So this makes this unit a little bit more interesting, I think, because we are dealing with this novel pathogen in the form of coronavirus right now. But anyway, it's easy to remember because when you first get sick, you need a doctor. You need an MD to help you, right? So that's easy to remember that those are the first to be expressed when you are first infected with a new to you pathogen. Now the other three here, so if we divide into what they do, essentially, A, D, and E are responsible for inflammation. A is a secondary exposure, so like the second, third, fourth, fifth times they're exposed to something. So is E. We already know that D is a first exposure sort of thing. Both of these are going to increase blood flow to an area to cause inflammation. IgE, this one is often triggered by allergies. So whether it's just like, oh, there's a lot of pollen in the air and I'm feeling really sneezy and my eyes are watering, or if it's something more serious where you end up with anaphylaxis, which is where your airways actually swell up and it's, get a lot of fluid buildup in there and it becomes difficult to breathe and you need to have an EpiPen with you. So if you're very allergic to something like peanuts or bee stings or sulfites, things like that can cause a severe allergic reaction. That's IgE that you have to thank for that. Now IgG, this does something called opsonization. This is making the antigen-antibody complex that we talked about before, where the antibody sticks to the antigen and makes it attractive to phagocytes. And then it can activate complement as well. And complement is a non-specific part of immunity. It's a bunch of plasma proteins that can be activated and do various things to help fight pathogens. Now, I do want to get into antigen presentation and programming naive T cells with the dendritic cells. We mentioned this briefly when we first brought up presenting antigens. There are cells that can present antigens and display a class 2 MHC, like monocytes, macrophages, B cells. They can display a class 2 MHC. But there's another type of cell, a dendritic cell, that can not only present a class 2 MHC and activate CD4 cells and helper Ts and all that, but they use this antigen presentation to program naive T cells. So naive T cells are cells that have not had a paratope programmed yet, and dendritic cells are essentially going to grab onto a parasite, or not a parasite, a pathogen. They're going to take it to a lymph node where you find these naive T cells, and they're going to program those paratopes with that viral epitope so that those newly trained T cells are no longer naive T cells. They are specific for that pathogen. So if this is a dendritic cell here, it has on it something called a pathogen recognition receptor. That pathogen recognition receptor recognizes pathogens. It grabs hold of the virus that's coming in, and you find these dendritic cells on mucous membranes all throughout your body. So those are points of entry for lots of pathogens. So here comes that virus into, say, your nose, and here's a dendritic cell and the mucous membrane there, and it sticks to that pathogen recognition receptor, and it enters the dendritic cell where it's getting digested then. So the dendritic cell is digesting this virus, and it displays that viral epitope on the class 2 MHC. Now as it's doing this, it matures, the dendritic cell itself matures and leaves the mucous membrane and migrates to the T cell, or to the naive T cells, which are found in the lymph nodes. It presents this antigen to those naive T cells and programs them with that particular epitope. So now those naive T cells have a paratope that is specific for that particular epitope. So that's how it should work. Now, HIV, so the virus that causes AIDS, is kind of a tricky little virus there. It binds to a very specific pathogen recognition receptor on the dendritic cell, and this is called the DC sign receptor. The DC sign receptor grabs onto HIV. HIV is pulled into the dendritic cell, and the dendritic cell presents that HIV antigen right there. So on an MHC2. And it's like, okay, I got this virus, and I'm going to go take it to the lymph node where I'm going to program some naive T cells. And it does that, and the T cells are like, yay, we're going to get programmed. But unfortunately, HIV has this way of, once the naive T cell binds with its CD4 marker, so it'll be CD4 naive T cells specifically, because HIV is just specific for the CD4 marker. HIV then gets into, it essentially uses a CD4 marker as a docking mechanism, and it uses it to wiggle its way into this poor naive CD4 T cell, and then there's HIV inside of this naive T cell, and it's using the machinery of the cell to make more HIV. So essentially what HIV has done is it's cut out a very key component of your immune system, which is the CD4 T cells and the helper Ts. So if you don't have helper Ts to be able to recognize HIV, then you can't do B cell sensitization either. You can't have helper Ts to help these B cells make antibodies for it, which is why HIV was, before they've found drugs that suppress it to the point of being pretty much non-existent. It was quite a deadly disease for many, many years. It would just destroyed the immune system. Now, luckily, they've got drugs that make it a completely manageable sort of thing. Okay, so something that is, actually let's make this a different video. Let's end here. We'll talk about this on its own video. This video is going a little long too. I don't want to kill you with 100 long videos. So we'll pause here and we'll pick up with the second video.